thank you to all the organizers for the invitation to come back. So I thought today I would tell you a little bit of an update on the DREAM talk I gave last year in Prague, and to tell you a little bit about the progress we've been able to make just in the last year toward hopefully getting closer to achieving that dream. Since there's only a couple people that were there last year, I thought I would just begin by telling you the motivation behind this particular dream and why we're interested in doing what we're doing and the specific approach we're taking. And then I'll tell you what we've been able to achieve to date. And so in large part, the project that I'm going to tell you about today is motivated by challenges associated with transporting gas molecules through water, which are relevant to a wide variety of energy and biomedical technologies. This includes things like electrocatalysis, where one wants to, say, undergo a chemical transformation of a small gas molecule, like reducing carbon dioxide to make a fuel, reducing nitrogen to make ammonia as a precursor to fertilizers, where one needs to get gas molecules from a gas phase, generally through an aqueous electrolyte, which can provide a source of protons and electrons, to an electrode where you have a catalyst that's going to convert that gas molecule. Gas transport in water is also relevant to a variety of gas separation challenges, including high-pressure acid gas separations like CO2 removal from natural gas. Also relevant to bioreactors, say where you have a living organism or cells that are producing some sort of bioproduct. You need to get oxygen into a large bioreactor where cells are generally suspended in some sort of aqueous solution or buffer. You need to get enough oxygen to those cells in order to be able to keep them alive so they can produce the product that you're interested in producing. And gas transport through water is, of course, relevant to a lot of biomedical challenges, situations where one wants to get oxygen either into the body or for ex vivo applications like organ preservation and some other things that we'll touch on later. The reason transporting gases through water can be quite challenging is because water is a very polar, strongly associated solvent that does not do a very good job of solubilizing gas molecules. So solubilities of most gases in water are about an order of magnitude lower than those of organic solvents or ionic liquids. Water molecules really like to interact with each other, and they don't like to move apart and to create space to accommodate gas molecules and to be able to dissolve them. You can really see this in particular in the solubility of oxygen in water, which is extremely low. Just to put this in context, if you think about a glass of water that's equilibrated with the air above it, so 0.2 bar partial pressure of oxygen in air under physiological conditions, so 37 degrees Celsius, the amount of oxygen that's dissolved in that water is about 0.2 millimolar, which corresponds to just a single oxygen molecule in every 8,000 cubic nanometers of space. So oxygen molecules in water under normal physiological conditions are very far apart, right? If you think about just this length scale in the context of the size of a lot of proteins and biological components, right? This is a very low density of oxygen. This is why early life forms were very limited in the size that they could grow, because they had to rely on passive diffusion of oxygen through seawater, and there's just not enough oxygen intrinsically present in water or seawater to be able to support large respiring organisms. The amount of oxygen that's present in water is about 40-fold less than the density of oxygen that's present in the air above it. So you have significantly more oxygen present in the gas phase. So this density of oxygen, of course, is not high enough to support life. And that's why over billions of years we evolved red blood cells and circulatory systems so that we can transport enough oxygen in order to be able to keep us alive. Over 84% of the cells in our body are red blood cells. Red blood cells don't contain a nucleus. They don't contain any other organelles. They're just packed full of hemoglobin, whose sole purpose is really to pack oxygen together at much higher densities so that we can get enough oxygen in an aqueous environment for cells to be able to respire. And so hemoglobin, of course, contains iron porphyrins, which chemically bind oxygen and allow oxygen molecules to come together to much higher densities. So red blood cells have about a 70-fold increase in the density of oxygen compared to the amount of oxygen that's dissolved in water, because we're co-localizing through this chemical bond between iron and oxygen molecules in a much smaller, over a much smaller distance than exists when they're dissolved in water. So as soon as the normal biological mechanisms for transporting oxygen fail, or if they're absent outside of the body, like when one is trying to preserve organs or to grow tissue, then one often runs into a lot of challenges. So we need about 230 milliliters of oxygen per minute in order to be able to stay alive. If your oxygen content in your blood decreases because your circulation stops because of something like a heart attack, or if you have some sort of lung injury, like you're choking, or you have acute respiratory distress syndrome, and you're unable to effectively get oxygen from the gas phase into your body, then this can lead to really rapid negative consequences that occur very quickly. The out-of-hospital survival rate for cardiac arrest is somewhere on the order of about 10%. And so there's been a lot of interest in trying to develop ways of effectively administering oxygen into the body, and there's been a lot of interest in trying to develop, in particular, ways of injecting oxygen into the body as kind of a bridge therapy and emergency medicine treatment to just buy an additional 1 or 2 minutes to keep oxygen levels high enough to avert some of these really bad side effects. And that's one kind of area in which we're interested in. It's really tempting to think that you're surrounded by oxygen in the air. It's really tempting to think that it shouldn't be that difficult to just get that oxygen from the gas phase into the body. You can actually directly inject oxygen gas into the bloodstream, but you're very limited in terms of the amount of oxygen that you can safely inject, to less than 10% of the amount of oxygen that we need to stay alive. And the reason for that is oxygen injected into the body will create air bubbles in your blood vessels, and these cause embolisms that can lead to blockage of blood vessels. Just a very small amount of gas bubbles in your blood vessels can have dire consequences. So just about a half milliliter of air as a bubble inside your blood vessels is enough to induce a heart attack. And if you have two milliliters of air in a brain blood vessel, that's enough to cause somebody to die. And so it's very hard, even though we're surrounded by oxygen, to be able to actually get it into the bloodstream. And so, again, there's been a lot of work that's done, some of which I'll describe, to try to develop ways of safely delivering oxygen intravenously for a lot of emergency medicine applications. But there's been a lot of interest in more broadly developing ways of transporting gases at high densities in aqueous environments to overcome a lot of the challenges that I alluded to. And again, this is not just true for in vivo applications, but also things like organ preservation, where the normal mechanisms for transporting oxygen are no longer present. Say you want to preserve a kidney for a transplant, you need to be able to take that kidney, which is submerged in some sort of aqueous medium or buffer, and keep it getting enough oxygen in order to maintain its quality and function for extended periods of time. And there's a lot of requirements that are placed on how you can store those organs to preserve them for transplants that could be alleviated by if we had more effective ways of getting oxygen through water. And so my group became really interested in thinking about new approaches to being able to transport gases efficiently and at high densities in aqueous environments. And if one wants to think about doing this, there's a few parameters that one needs to really carefully consider. One, whatever your vector for transporting gas molecules is, needs to be able to store a high density of gases. Right? We want to be able to co-localize a large amount of gas molecules in an aqueous environment so that we can transport a high flux of gas. Whatever our vector is for transporting gas molecules needs to be able to bind that gas reversibly. Just like hemoglobin, we need to not only be able to store the gas, but we need to be able to release it where and when it's needed. In some cases, we may want to be able to release that gas really quickly. In other cases, we may want that gas to be released slowly. Ideally, we'd like to have some degree of control of the kinetics for gas release. If we're thinking about biomedical applications, then we need to be concerned with the toxicity of whatever our vector for transporting the gases, and we need to make things that are biocompatible. Much less important if we're thinking about electric catalysis or more energy-related applications. And so a lot of work toward developing artificial gas carriers that could function in aqueous environments, and in particular artificial oxygen carriers, really dates back to the 1960s, when there was a lot of interest in developing artificial blood substitutes to deal with shortages of donated human blood. And this was really accelerated in the 1980s with the HIV crisis, when there was a huge shortage of donated blood. And so there's been two kind of primary strategies that people have explored over the last 50 or 60 years to develop these kinds of artificial blood substitutes. One is to directly mimic what nature already does and have some sort of reactive iron center that can undergo an electron transfer reaction with oxygen, chemically bind oxygen to increase its density. This includes things like cell-free hemoglobin or more inorganic molecular heme mimics, where one is trying to kind of replicate the chemistry that exists within hemoglobin and in red blood cells. It turns out hemoglobin itself, once you take it outside of a red blood cell, is actually very toxic and can induce a lot of kidney damage and can be really difficult to make cell-free hemoglobin that is both not that cytotoxic or has high biocompatibility, where also you get the binding between oxygen and these iron centers correct so that you can actually easily release it when it's needed. Red blood cells are a very finely tuned and kind of regulated environment, and outside of that, it's difficult to get this to function properly. So there's never actually been in the US an FDA, Federal Drug Administration, approved hemoglobin-based product for transporting oxygen. And I believe that's true throughout the world as well. The other approach that people have looked at is to take high oxygen solubility solvents, things like perfluorocarbon liquids, which I'll talk a bit more about as a second, make them into emulsions in water and transport oxygen by physically dissolving it at high densities. So here, these are solvents that can, liquids that can absorb much higher quantities of oxygen than water. These basically increase the density of oxygen through physical dissolution rather than chemical binding mechanism of oxygen. So these have actually had the most success clinically, so I'll just describe a little bit more what's going on in some of these solutions. So perfluorocarbon liquids, as I mentioned, have much higher oxygen solubilities than neutral organic solvents, as well as than water. So about an order of magnitude more oxygen can be dissolved in a lot of perfluorocarbon liquids compared to water, and that's because perfluorocarbon molecules are very weakly associated, unlike water, and the thermodynamic penalty for dissolving a gas inside the perfluorocarbon is significantly less. And so that's why these were of interest for transporting oxygen. Carbon-fluorine bonds are also really strong, and the body generally doesn't really know what to do with them. And so perfluorocarbons tend to be relatively biocompatible in most cases. And so if you just take a pure perfluorocarbon liquid, this is, I think, perfluorodecane, you can actually submerge a mouse and its lungs will fill with liquid, but it won't drown because there's enough oxygen that's intrinsically present in the perfluorocarbon liquid to keep the mouse alive. So this is a famous experiment from the 1960s with a mouse that was ventilated with liquid perfluorocarbon solvent, just equilibrated with air through passive diffusion, that stayed alive for 4 hours and remained alive even when outside the perfluorocarbon liquid. So this goes to show how much oxygen is there in this kind of liquid compared to water. So as an artificial blood substitute, one doesn't use a neat perfluorocarbon, but one takes droplets of perfluorocarbon liquids, coats them in some sort of surfactant because they're not inherently miscible with water, so things like a phospholipid or even different types of proteins, to form generally micron-sized droplets of perfluorocarbon liquids that are dispersed within water to form these kinds of emulsions. So still, though, even though these solvents have much higher oxygen solubilities than water, the amount of oxygen that you can actually store even at relatively high concentrations of perfluorocarbon, over 60 volume%, is relatively modest, and it's very hard to make a perfluorocarbon emulsion that has the same oxygen carrying capacity as blood. These are two commercial perfluorocarbon emulsions that have gone through clinical trials for a wide variety of different kinds of oxygen delivery applications. The other issue with these emulsions is that perfluorocarbon molecules are really good at dissolving oxygen because they're very weakly associated, but that causes a lot of issues for the stability, particularly long-term stability of these emulsions. So droplets of perfluorocarbons tend to coalesce and grow larger in size over time, and that creates challenges as you get to larger and larger droplets, particularly if you want to do anything that involves injecting them into the body. There's also been issues in clinical trials with organ retention of perfluorocarbons and just the amount of oxygen or amount of perfluorocarbon that you really need to be able to dose to have a meaningful impact, leading to bad side effects and significant toxicity. So there's been interest in thinking about other approaches that one could use that could circumvent some of these challenges. There has been one perfluorocarbon emulsion that was approved for use in corneoangioplasty surgery in the US in the 1990s, but its approval was actually withdrawn because of a lot of these challenges. So my group became interested in alternative strategies for concentrating oxygen and other gas molecules in water, and we were particularly motivated by some of our experience working with porous materials and thinking about adsorption at surfaces as a way of concentrating gas molecules rather than dissolution in a weakly associated solvent or chemically binding oxygen at a reactive metal center. And so just for those that don't think too much about this, basic adsorption of a gas molecule on a surface, you have a gas, imagine, say, oxygen, represented by these spheres that exist at some density in the gas phase. You put the gas in contact with really any surface. If there's any kind of attractive interaction, even weak ones, between that surface and these gas molecules, then you're going to increase the density of gas molecules that are present on that surface because of these attractive interactions. These can be very weak. You don't have to really change the gas molecule itself, so there doesn't have to be electron transfer or chemical binding of the gas at the surface, but just very weak 5 to 10, even 15 kilojoules per mole van der Waals interactions that polarize slightly the electron clouds around gas molecules can have a big effect in terms of increasing the density of a gas that's present at a surface. And so when you think about surface adsorption, there'll be an increased density of gas that's present at a surface compared to the bulk phase gas density. And so this is true for a large surface. It's also true for the internal surfaces of materials. So if you imagine folding up a surface to sub kind of nanometer dimensions inside a material, you can have a large amount of surfaces that are able to interact with gas molecules that can really significantly increase the local concentration of gases. And this is why microporous materials like the metal organic frameworks that Carl very nicely introduced in his presentation today are very good at concentrating gas molecules to high density, and why they've received a lot of attention for a lot of gas storage and separation applications in the solid state. So microporous materials are materials that tend to have pore sizes that are below 2 nanometers. And again, they have a large amount of surface area inside them that can have attractive interactions with gas molecules and can significantly increase the density of gas that's present compared to the bulk gas phase. Just for some perspective, some of the highest surface area metal organic frameworks now, in just about 2 milliliters of material, you can have surface areas that are on par with those of an American football field or even a European football field as well. And this is a huge amount of surface and a very tiny volume of material. And if you have these attractive interactions between gas molecule and the surface, you can just imagine the amount of gas that you're going to be able to compress and compact inside a material under moderate conditions. And this is why these are very powerful materials for storing gases. To be a bit more quantitative about this, this is just a comparison of the density of oxygen that's present within a porous material, including the amount of space taken up by the porous material itself. At 1 bar in ambient temperature on the left side, and on the right side is the density of carbon dioxide for 2 kind of run-of-the-mill representative metal organic frameworks and 1 zeolite, which I'll talk a bit more about in a second, compared to the bulk gas density and compared to perfluorocarbon liquid and compared to water. And what you see is orders of magnitude enhancement in the amount of gas that you're able to store in these materials because of the really high internal surface areas, even for materials that interact very weakly with gas molecules. And so we were excited about thinking about the potential of using these classes of microporous materials for overcoming gas transport limitations in aqueous environments, seeing if we could really take advantage of the fact that we're able to concentrate gases to such high densities to be able to transport gases and water. So the problem that you run into if you want to think about doing this is as soon as you take most microporous solid materials that again have really high internal surface areas, really good at absorbing gas molecules, as soon as you put them in water, water will tend to fill those pores, right? Water is going to be smaller than most gas molecules that you would want to absorb. You flood the pores, you remove all of this internal surface area, and you wind up generally with an aqueous solution that actually dissolves less gas than if you didn't have the porous material present. And so most porous materials are not actually that useful for transporting gases in water. And so we wanted to find a way to get around this and a way to preserve actually the porosity of a microporous material in water and see if we could leverage that for transporting gases effectively. And so we were inspired in part by 2 things, one from biology, and that's the fact that there's a lot of proteins that exist naturally that have really large cavities that are not closed. They're open to the aqueous environment, but the cavities themselves are actually dry. So water is accessible to the cavities, but water doesn't actually go inside the cavities because it's thermodynamically unfavorable for it to do so. This can be both enthopically and entropically unfavorable. So entropically unfavorable because water molecules have less degrees of freedom when they're confined inside these hydrophobic pockets than in the bulk phase, and enthopically unfavorable because water-water interactions are stronger than water interactions with these hydrophobic surfaces of these cavities. It's also known that if you take hydrophobic porous materials, think like activated carbon, and you put them in contact with bulk water, it can often require significant amounts of pressure in order to force the liquid water into the pores of that material. So for activated carbons and for some really hydrophobic zeolites, it's known that you have to put anywhere from 200 to 800 bar of pressure in order to force water inside the pores of a hydrophobic solid. And so this made us think that there was kind of a thermodynamic approach that might work for preserving porosity of microporous materials in water and preserving their gas accessibility. And so in particular, what we wanted to do was to design microporous nanoparticles that had really hydrophobic internal surfaces such that it would be thermodynamically unfavorable for water, liquid water, to go inside the pores, just like in the two examples I just discussed. But it would remain thermodynamically favorable for gas molecules to go in the pores, and they would still be accessible to gases. The problem is if you take most hydrophobic materials and you try to disperse them in water, that usually doesn't work really well, right? They tend to clump up, aggregate, settle out of solution, and that's not going to be particularly useful for trying to actually transport a gas through water or make an artificial blood substitute or anything like that. So we wanted to make the external surfaces of these particles really hydrophilic so that you could form really nice uniform dispersions in water. So a relatively simple idea that we've been able to demonstrate over the last couple of years. So the first type of material that we used to demonstrate this concept would actually work was a zeolite. And so just a brief introduction of zeolites for those of you who are not familiar. These are materials that are generally composed of silicon or aluminum cations bridged by oxygen anions. So you have tetrahedral silicon or aluminum units that are linked together in various ways to form three-dimensional porous structures. The materials that we work with so far are mostly pure silica zeolites, so they contain just silicon and oxygen, overall chemical formula SiO2, and they're neutral frameworks. The particular zeolite that we started working with is known as silicite 1. This is a rather old zeolite that was first synthesized in the 1970s. The reason we were interested in this particular zeolite was, one, that it has quite a high surface area. It's about a third empty space, so a very porous material. But even more importantly, these silicon-oxygen bonds are quite nonpolar, and the internal surface of this zeolite looks very much like that of an activated carbon, so it's very hydrophobic. The internal surface of this material contains a porous network of about 0.5 nanometer pores that are interconnected in 3 dimensions. The other reason we were really excited about this material was that its external surface, the external surface of pure silica zeolites, are terminated in silanol groups, and these silanol groups can be deprotonated in water. And so what you wind up with is an external surface that's intrinsically hydrophilic. So you have a hydrophilic external surface and a very hydrophobic internal surface. And so a very talented graduate student in my group, Dan Erdesi, spent a lot of time optimizing the synthesis and activation of these pure silica zeolite nanoparticles so that we can make them with relatively uniform sizes and be able to actually disperse them in water. These zeolites are made by combining a silica precursor, in this case tetraethyl orthosilicate, with some type of organic structure directing agent, in this case tetrapropyl ammonium hydroxide, which basically the silica crystallizes around the organic cation, which templates the pores. The challenge with doing this is that you then need to remove the organic structure directing agent, which is usually done by heating at really high temperatures, but you need to be able to do that without irreversibly aggregating the nanoparticles in the solid state, and that requires a fair bit of optimization, which Dan was able to do to be able to make nanoparticles of a variety of different sizes. This is one example here, but we can make anywhere from about 50 nanometers to as large as 300 or 400 nanometer size nanoparticles that after you spend some time purifying them and figuring out how to activate them can form really nice colloidally stable dispersions in water. These can be really, really stable dispersions where we don't observe any signs of settling or aggregation over periods as long now as like 6 to 8 months. These are translucent solutions that at moderate concentrations and small particle sizes really look like water. So the first thing we wanted to do is to actually convince ourselves that in these solutions that look like water, that we actually can have 5 or 10% empty space, and that the pores of these zeolite nanoparticles, even when you're in a nanocrystal that's surrounded by water, actually do remain completely dry. And so the way we did that initially was just by a relatively simple experiment where we looked at the density of these dispersions as a function of concentration, and then density of a solution that contains nanoparticles with water-filled pores is going to be higher than the density of a solution that contains air-filled or dry pores. And so what we found is that in our Silicalite 1 dispersions, we see densities that are very consistent with what we would predict for completely dry pores. And this was in contrast to what we see if we use a less polar solvent like ethanol instead of water. We would not expect it to be thermodynamically unfavorable for an alcohol to go in the pores. We would expect that to be thermodynamically downhill. And we see densities that are consistent with having solvent-filled pores. If we use a hydrophilic zeolite instead of a hydrophobic zeolite, so this is just another zeolite that contains a mix of aluminum and silicon cations, and its pores are charged, and so they're quite hydrophilic. Then we see densities in water that are consistent with having water inside the pores. So this convinced us that the pores were likely dry inside these colloidal dispersions. So the next question was, are they accessible to gas molecules? And can they remain dry even when we degas the solutions and put them under vacuum? So 2 other graduate students, Malia and Joy, and my group spent a lot of time working out how to do direct measurements of gas uptake in these degassed colloidal dispersions. So we put them under vacuum, we remove all the gas from the dispersion, and then we expose the solution to gas at different pressures, and we directly measure how much gas is absorbed within the solution. And so what we see is in our hydrophobic zeolite dispersions, significant enhancements in the amount of gas that's taken up by these aqueous solutions, both for oxygen and carbon dioxide, compared to a dispersion of a hydrophilic zeolite and compared to water. And so this is directly confirming that the pores of our materials are not only dry, but they're accessible to gas molecules and still able to absorb gas molecules even in this kind of dispersion. And so this got us really excited. We also worked closely with some collaborators at Northwestern University, particularly a group of Monica Olvera de la Cruz, to do some molecular dynamic simulations to actually visualize exactly what's going on when we're doing these kinds of experiments. And a really talented research scientist, Felipe, and her group built a model of a 10 nanometer Silicalite 1 nanoparticle, which you can see right here, which is surrounded by liquid water and in contact with a bulk gas interface. This is just oxygen gas at about a pressure of 1 bar when the simulation starts. So it doesn't matter if we put water molecules inside the Silicalite 1 nanoparticle at the beginning or not. If they're inside rapidly within a nanosecond, they're out of the Silicalite 1 nanoparticle. But what you'll see as we run the simulation is that oxygen molecules are rapidly kind of moving around, and the density of oxygen, the concentration of oxygen inside the Silicalite 1 nanoparticle will increase over time, while the density of water remains constant. Water stays out, but oxygen goes in and is absorbed. What you can also see in the simulation is that the interactions of oxygen with the internal surfaces of the zeolite nanocrystal are extremely weak. So these are on the order of about 10 to 15 kilojoules per mole, about similar to a hydrogen bond. So these are very weak van der Waals interactions. So oxygen is rapidly diffusing within this material and rapidly exchanging with the surrounding aqueous environment. And so this is really important for evaluating the reversibility of gas storage within these materials. Gas exchange with the surrounding environment we predicted would be very fast because gas uptake is so weak within these materials. So it's really this high internal surface area that's leading to this enhancement in gas density. It's not strong interactions with gas like or with oxygen like in hemoglobin. So then one of the next things we wanted to do was demonstrate not only that we could absorb gas molecules, but that we could actually confirm that we could release them and to evaluate, at least to some extent, the kinetics of gas release. And so to do that, we took oxygenated solutions, so oxygenated dispersions of our porous materials, and injected them into deoxygenated water, and then used an oxygen electrode to quantify exactly how much oxygen is released. What happens here is that, say we equilibrate at 1 bar of oxygen. Here we have 0 bar of oxygen, so there's a partial pressure or concentration gradient between our oxygenated solution and our deoxygenated solution, and that drives the desorption of oxygen from the particles into the surrounding solution. And so we can use an oxygen electrode to quantify exactly how much gas is released as we inject different quantities of these solutions. And what we find is that the amount of gas that's released is very consistent with what we have measured directly with our gas uptake measurements and what we would predict based on the known adsorption properties of these materials. What we also found was that gas release was really fast, faster than we could measure in the time scale of this experiment, which is consistent with what we see in molecular dynamics simulations. So one of the reasons we got really excited about this approach was just by looking at the amount of oxygen that we were actually able to store within these solutions. And so to put this in context, just comparing here the oxygen carrying capacity of our solution, silicate 1, these zeolite nanoparticles, at a relatively low concentration, so this is less than 4 volume percent, we're matching the oxygen carrying capacity of blood and far exceeding what can be achieved with perfluorocarbon emulsions at much higher concentrations. And so, this solution is 96% water, but we're concentrating oxygen to the same density as what you have in blood. If we go to higher concentrations of these zeolite nanoparticles, then we can match the bulk gas oxygen density in water. So you have the same overall density of oxygen in your aqueous solution as you do in the gas phase above it. If you play with particle size, you can get to quite high concentrations of solutions at relatively moderate viscosities, so viscosities that are comparable to those of blood. And you can reach much higher concentrations of solutions. So I think we're up to about 50 volume percent now, and you can get oxygen carrying capacities that far exceed the gas density. So this got us excited about kind of using these aqueous solutions for a variety of different applications where one is really limited by how much gas you're able to transfer through water, both on biomedical applications and energy applications. So we did one kind of proof of principle experiment initially where we showed that we could take these oxygenated silicate 1 solutions, we could inject them into deoxygenated donated human blood and do normal kind of blood gas analysis measurements, just like you would get if you see a doctor, to quantify change in oxygenated hemoglobin and measure how much oxygen is released. We found that the oxygen carrying capacities that we measured doing these blood gas analysis measurements are again in line with what we measured using these other techniques in pure water. And so this got us excited about exploring biomedical applications with these solutions. Before getting back to that, I'll just kind of mention some ways in which we've been able to generalize this approach beyond just metal organic frameworks to other classes of porous materials. Since Carl did a really nice job of introducing metal organic frameworks, I'll just briefly reiterate that these are materials that contain small inorganic clusters or metal cations that are linked together by different kinds of bridging organic ligands to form three-dimensional porous materials. The reason we were excited about extending this approach to metal organic frameworks is that there's something like 90,000 metal organic frameworks that exist currently in the literature compared to about 240 zeolites. And these materials exhibit much greater structural and chemical diversity than all inorganic materials like zeolites. So when one wants to tune biocompatibility, gas uptake and release properties, there's just a lot more that you can do with the MOF platform compared to other platforms of porous materials. The problem you run into with most hydrophobic metal organic frameworks are, one, it's kind of hard to make metal organic frameworks that are hydrophobic enough to exclude liquid water. And two, any metal organic frameworks that are hydrophobic enough tend to have very hydrophobic external surfaces and tend to just aggregate in aqueous environments. And so we spent a lot of time really over the last 1 to 2 years developing ways of externally functionalizing selectively the external surface of metal organic framework nanoparticles to make those surfaces hydrophilic. And one of the challenges that you have in doing that is that you need to be able to put surface ligands on the external surface of a porous material without blocking access of gas molecules to the internal pores. And you need to make sure those surface ligands don't go inside your pores and remove all of your porosity. And so doing that can be quite challenging, but there's a couple of different approaches that one can take, some of which were alluded to by Carl. You can think about covalently attaching different surface ligands like modulators to the external surface if you have some way of ensuring that they remain on the external surface. You can also think about non-covalently adsorbing large macromolecules that are too large to fit inside the pores, things like polymers and proteins that can make a hydrophobic surface much more hydrophilic. And so we've done both approaches. I'll just give you one example of a hydrophobic metal organic framework. In this particular case, this is a zinc or cobalt-based MOF that contains 2-methylimidazole as the bridging organic ligand. Has a structure very similar, actually, to some zeolites. Quite porous, has a really high surface area. We developed a new kind of covalent strategy for functionalizing its external surface with ethylene glycol-like oligomers that takes advantage of the fact that we believe there's 2-methylimidazolate ligands that are present on the external surface of this material that are not present because they're bridging in the inside of the material. And these are basically kind of reactive handles that are capable of ring-opening epoxides and allowing us to selectively functionalize this external surface and make it very hydrophilic. We've also spent a lot of time looking at how different proteins and polymers interact with this external surface to try to develop design principles for making these surfaces hydrophilic while still preserving porosity in aqueous environments. And so Joy and a postdoc, Chris in my group, have been able to demonstrate this strategy works for some different MOF nanoparticles where we can make really stable colloidal dispersions of these particles with either covalent or non-covalently functionalized hydrophilic external surfaces that contain completely dry pores that are accessible to gas molecules. Just give you a couple examples of some of the interesting kind of more fundamental things that we've observed so far. One, proteins interact very strongly with the external surfaces of these MOF particles, which can be useful for making them hydrophilic and allowing one to form dispersions. Can potentially be problematic for biomedical applications if it's going to trigger an immune response. And so one of the things we want to do is just better understand how proteins interact with this external surfaces so that we can either use that to bring new properties to our dispersions or try to prevent it through different functionalization approaches. And so this is just an example of bovine serum albumin, which sticks really strongly to the external surface of these ZIF particles. Some of the interesting things we found is that depending on the identity of the protein, so both its surface charge and size and structure, we see very different properties that can be induced in our dispersions. So these particular class of metal organic frameworks, these ZIF metal organic frameworks, are actually not that stable in water, and at low concentrations they will dissolve. So this gets back to some of Carl's kind of dynamic metal ligand bond arguments. And at low concentrations, you shift the equilibrium toward having water bind the metal center rather than your bridging organic ligand. And a lot of these will dissolve in water at low concentrations over time, which can be useful for some biomedical applications where you need to be concerned with how particles will be cleared, and is less useful for other applications. But more fundamentally, we're just curious to try to start understanding why do different proteins induce very different changes in the actual stability of a lot of these MOF particles. And so what we find is that if you go from, say, casein to a positively charged protein like lysozyme to a negatively charged protein like bovine serum albumin, we see very different behavior in terms of the stability of the MOF particles. So with BSA at low concentrations, these MOF particles dissolve within a matter of minutes. With casein, we see particles that are very stable for much longer periods of time. And so this is something we're still trying to understand. We don't think it's really an effect of how much protein is absorbed on the external surface of the particles, but more an effect of how the proteins are kind of packing together and altering the kind of solvation cell or hydration shell of the particles, and how they're controlling access of water to the pore surface. And this is something, again, we're still working to understand. We also see some really interesting phase behavior depending on the size of the particles we're working with and the concentration of the particles, where in some cases with certain proteins, we see what looks like a liquid-liquid phase separation over time, where we see liquid region that contains a high concentration of our MOF particles and protein, and one that contains a very low concentration of MOF particles and protein. These particles, MOF particles, are less dense than water, so the particles actually rise to the top. If you put alcohol in the solution, then they'll sink back to the bottom. But we see this kind of phase separation behavior, again, at some concentrations and with some particle sizes for certain proteins, and we don't see it at all for other proteins, regardless of the size of the particles or the concentration. And again, trying to really understand what's dictating this different kind of phase behavior is something my group is working hard to understand, and something that we're really interested in. We can also see some really interesting things with looking at how polymers interact with the surfaces of these particles. Carl, again, kind of alluded to this in thinking about mixed matrix membranes and how one controls the interface between polymers and MOFs is really important for that context. For our purposes, we were interested in using polymers both to make particles more biocompatible and to make them more dispersible and stable in water. And so one of the most common strategies for making any kind of nanoparticle more biocompatible, a strategy that was actually used in the COVID vaccines, is to add PEG. PEG generally cuts down on protein absorption and tends to make things more biocompatible. Problem with PEG for these MOF nanoparticles is that PEG is quite small, and it's thermodynamically favorable actually for PEG to go inside the pores of our material. And so PEG rapidly intrudes into the material's pores. You lose all your porosity, you lose all your oxygen carrying capacity, and so it's not an effective strategy for making these materials dispersible in water. So Chris, a postdoc in my group, looked at different block copolymers that can still contain PEG, but contain much more sterically bulky blocks, including polystyrene and polyfluorovinyl pyridine. And what he found is that with certain block copolymers, particularly ones that are quite bulky, we can actually preserve a lot of the porosity of our particles, even with the same amount of PEG present. So basically, we think that these kind of blocks are serving as kind of anchors that are holding some of the polymer on the outside and preventing PEG from getting inside the particles. But again, this is something that we're still really working to understand, and in particular thinking about the thermodynamics of these interactions of polymers with the surfaces and internal pores of these materials and kind of how we can manipulate that. We do see really interesting differences in the stability of these particles as well. So you'll notice that even with polyfluorovinyl pyridine, which contains these pyridine units, which could actually coordinate directly to metal centers on the framework, polystyrene, which cannot, we see pretty similar behavior in terms of the porosity of the solutions, but we see very different behavior in terms of their stability at low concentrations. So polyfluorovinyl pyridine solutions are very stable at low concentrations, whereas polystyrene and the pure ZIF particles will rapidly dissolve. And so we think that the pyridine groups are kind of binding to reactive metal sites on the external surface and preventing water from binding, and that's enhancing its stability in water. So again, just kind of demonstrating some of the things that we can play with here to tune the properties of our dispersions in different ways. So we're really excited about this kind of as a platform for being able to address a variety of challenges with transporting gases in aqueous environments and really leveraging these high gas carrying capacities and the tunability of these materials to design solutions that are kind of optimized for different applications. And so in the last couple minutes, I'll just give you a couple examples of some of the things we're thinking about along that front. For biomedical applications, of course, we need to be concerned with biocompatibility of our materials, which is not something we really cared at all about in these kind of initial proof of concept demonstrations. The pure silica zeolite nanoparticles tend to not be very biocompatible. These silane surfaces are really good at interacting with cell membranes and ripping them apart. So they'll generally have quite high cytotoxicity. This is known from a lot of literature related to developing silica nanomaterials for drug delivery applications, which is quite advanced. So different zeolite nanoparticles can have very different cytotoxicities depending on their structure. This is an example of mesoporous silica nanoparticles that were investigated for drug delivery applications. Again, in their native form, when you have a high concentration of silanol, it's very cytotoxic, so to red blood cells, induce a lot of hemolysis. But if you covalently functionalize the external surface with PEG, you can basically significantly enhance their biocompatibility and basically remove all of this hemolysis. And so this is just a demonstration of the fact that what's on the external surface of the particle is generally governs its biocompatibility. What's on the internal surface for us is what governs its gas absorption and release behavior. And so we can independently manipulate these different aspects of particles. Similarly, for metal organic frameworks, there's a lot that's not really understood about their biocompatibility and their behavior in biological environments. And a lot of this comes down to the specific metal organic framework, the size of the particles, what's on its external surface, and how exactly it's being administered and what it's interacting with. And so there's a lot to kind of understand here that we're working to understand. There's certain applications where biocompatibility actually doesn't really matter. And so one of these, a brief example I'll give, is related to different types of therapy for cancer, where you can imagine directly injecting some sort of therapeutic into a tumor. And since you're injecting into a tumor, you don't really care about how biocompatible what you're injecting is, because you're generally trying to kill the tumor. And so one challenge with a lot of different cancer therapies is that tumors tend to be very hypoxic because the cells in a tumor are rapidly proliferating and you have very leaky blood vasculature. And so if you look at the partial pressure of oxygen in a lot of tumors, it's much, much lower than in healthy tissue. And this actually creates a lot of challenges for different types of cancer treatments, in particular radiation treatment, where you're using ionizing radiation to induce a radical on DNA. This radical that you make on DNA is kind of fixed by oxygen. So you react with oxygen. This triggers a DNA double helix strand break and can lead to cell death. If you don't have much oxygen present, though, this ionization can actually be pretty quickly repaired by the body. And so the effectiveness of radiation treatment is basically directly related to how much oxygen is present in your tumor. The more hypoxic your tumor is, the more radiation you need to induce a given amount of cell death. And so there's a lot of groups that have looked at ways of reoxygenating tumors prior to radiation treatment. And so we did kind of an initial proof of principle experiment where we demonstrated without really any optimization that we could inject our hydrophobic zeolite nanoparticles in water prior to administering radiation for a mouse tumor model, and show that we could significantly slow the growth of a tumor compared to control experiments by providing the source of oxygen. If we inject the zeolite with just nitrogen rather than oxygen, we see no change in how quickly the tumor is growing. So there's other applications too where biocompatibility constraints are kind of less rigid than for in vivo applications, and these are things that we're actively exploring as well. So looking at using these as solutions for increasing the rate at which you can transport gases in, say, aerobic bioreactors, or also for three-dimensional cell culture and growing tissues and things like that, where you need to be concerned just about a small subset of interactions between your vector for delivering gas and different biological components, rather than in vivo applications where you need to be concerned with a lot of things. One kind of non-biological example that I'll end with that gets at kind of overcoming some of these gas transport limitations is some recent work that we've done in collaboration with one of my colleagues in inorganic chemistry at Harvard, Dan Nocera, which was to look at using these solutions as electrolytes for electrocatalysis applications, where similarly we're trying to deal with challenges of transporting gas from a bulk gas phase through an aqueous electrolyte to an electrocatalyst. And our idea was that having these porous solutions with high gas densities, these would basically serve as kind of vectors for transporting gas quickly to the electrode surface, and could overcome some of these mass transport limitations. And so we tested this with a simple reaction, so the oxygen reduction reaction, where we're converting oxygen into water. This reaction is done commonly in fuel cells. And in conventional electrochemical setups, the reaction is very mass transport limited by the low solubility of oxygen in water. And so that dictates the current density that you can achieve under normal kind of conditions. So there's a platinum catalyst on the electrode that serves as the catalyst for ORR. And so what we found, working with a postdoc in Dan Nocera's group, Agnes, was that as we increase the concentration of our silicate-1 hydrophobic zeolite particles in these aqueous electrolytes, we see significant enhancements in the current density that we're able to achieve because we're removing a lot of these mass transport limitations for oxygen. So these solutions effectively serve as an oxygen buffer, and when oxygen is being reacted at the electrode surface, we have nanoparticles full of oxygen nearby that are rapidly able to release and replenish that oxygen. And so this allows us to get to much higher current densities. The other thing we were really excited about was that as we are able to achieve these higher current densities, we see a shift to lower potentials or higher over potentials in where we actually reach this kind of plateau current density. And through some modeling efforts, what we were able to understand is that we are actually approaching a region where we're limited by the intrinsic performance of the catalyst rather than limited by mass transport of oxygen through water. So under normal kind of conditions, we're really just limited in terms of the current density we can release by oxygen solubility and mass transport of oxygen. But under these high oxygen electrolyte conditions, we're more limited by catalyst performance. And so this is allowing us to kind of get a direct measure or evaluation of the intrinsic performance of this particular platinum catalyst in a conventional electrochemical setup, which is something that is really challenging to do. So we think there's a lot of opportunities here for exploring how these kinds of solutions can be used to provide new ways of doing things related to electrocatalysis as well. So I'll end there and just thank everyone in my group that contributed to this research. I've been fortunate to work with some really talented undergraduates and graduate students and postdocs during my time at Harvard, as well as some really excellent collaborators. Thank you again for the invitation to come back here. It's been a real pleasure, and I'm happy to answer any questions that you have.
Thank you, Jarad. It was a nice presentation about how answering to very simple questions can lead to far-reaching consequences, right? That's, so I open the discussion. Do you have any questions? Yes.
Fantastic talk.
Thanks.
I have one question about the possibility of other gases, because the whole lecture was focused on oxygen, and rightly so. This is a very important topic. But do you think that, let's imagine that you equilibrate with a mixture of few gases, like nitrogen, carbon monoxide, carbon dioxide. What will happen then? Or you tried already, or if not, what do you think will happen?
Yeah, so gas mixtures are certainly interesting. So in a lot of the experiments we do, we actually are using a mixture of gases. So we're often equilibrating with air. And so we have nitrogen and oxygen that are both inside the porous material. Most of these porous materials are not really selective for nitrogen over oxygen or oxygen over nitrogen. So they will adsorb to a similar extent, and it's just kind of governed by their relative partial pressures. If you have gases like carbon dioxide, and CO2 is much more polarizable than nitrogen or oxygen, and so it will adsorb much more strongly in nearly any porous material. And so these materials are selective for CO2 over oxygen, but the amount that's present will depend on the relative partial pressures. And so depending on what kind of application you're interested in, thinking about gas selectivity and mixtures of gases may be important. And so for gas separations, that's something we're really interested in. For biological applications, it is something that you have to think about, because you need, generally, if you want to transport oxygen, your vector needs to not only be able to deliver oxygen, but also remove CO2. And so thinking about that kind of interplay is really important. You also have to think about NO, and you don't want to remove a bunch of NO from the body. And so thinking about pre-equilibrating with NO and all these kinds of things are things that one has to think about.
Thanks a lot.
Questions, comments? Actually, you mentioned that this PEG molecules can enter the nanopores, but it is sufficient to make the radius of gyration a little bit larger than the pore size. The pore size was 5 angstroms. So, you know, they would never ever get in because of the partition coefficient would be 0. You just...
Yeah, for the zeolite nanoparticles where the pore size is on the order of 5.5 angstroms, then using larger particles definitely works. PEG still goes into those materials.
What was the mass of the PEG?
So we've used everything from molecular weight of 1,000 all the way up to, say, 30,000. And in all cases, we see, so it doesn't have to, the other thing is it doesn't have to go all the way in order to fully remove the gas capacity, right? So even if PEG just kind of goes a little bit in, if enough of it goes a little bit in around the surface, even if you still have a hollow empty core, it won't be accessible to gas molecules anymore. And so we can't really distinguish between how much.
It must be at the surface. You know, it does not get in because the partition coefficient would be like, with 30,000 would be 10 to minus 76.
Yeah.
So that would be outside. And so somehow it is at the surface. It does not enter inside. So I'm pretty sure of that. I can
for silicate 1, actually, if we get up to 30,000, we actually don't see it go in. You're right. And if it's at 1,000, it does go in. For the MOF particles, the pore size is quite a bit larger, and the pore windows are much more dynamic. And so there, we're confident it does go in because we can actually see it through small
unless you have a kinetic trapping. Unless the pores open sufficiently to kinetically trap the polymer.
So in the MOFs, the pores are closer to around 8 to 9 angstroms in size, and there we actually are confident that they're going
in. Just a technical question. How you measure such a tiny change of density in nanosecond time scale?
So the nanosecond timescales was all simulations. So that is all done through molecular dynamics simulations. So any of the kinetics measurements we've done, our time resolution is on the order of seconds. And that's why we're not able to actually experimentally evaluate kinetics yet. Yeah, good question.
I wonder how does your method of capturing oxygen compare with the photochemical process. You can easily generate singlet oxygen, and then it reacts, for instance, with anthracene. Then you heat and you recover oxygen.
Yeah. So that approach to delivering oxygen has often been what's used for, particularly for a lot of these tumor treatments, as a way of generating oxygen. You can run into issues with just the fact that you have something that's really reactive, and so generating radical species can be a problem depending on the particular application. So for certain applications, that's a viable strategy. I think if you want to make an artificial blood substitute or things like that, it's hard to envision how you would be able to kind of...
Well, you can use porphyrin, which is actually quite human-friendly.
Yeah, so there's been a lot of efforts to try to do it with porphyrins that have run into a lot of challenges with either biocompatibility or just getting the oxygen release correct. But in principle, you know, it's a viable strategy, and you can achieve similar densities of gas in a lot of cases, but you have to be able to get that release correct, so a different mechanism.
Okay, it's nice seeing the dream coming true after a year. Congratulations. To this thing, you already mentioned the DNA damage in the tumor, so I worked a little bit on this. So this is the usual picture that they show, that the radiation actually oxidizes DNA, but that's only a minor mechanism. Dominantly, you're oxidizing everything around it, like water. So, and it's the OH radicals and other radicals. So I wonder what's the role of the oxygen there, but probably the pathway to creating these oxygen radicals, the dominant pathway is not through the DNA directly, because you just have such a small amount of DNA. So.
Yeah, that makes sense. I think it does seem pretty clear that the amount of oxygen plays a role, and so it may be more complicated in that the amount of oxygen is related to the amount of other reactive species that you're generating. Yeah.
So OH radicals are thought to be the dominant primary radicals, but then they radicals.
Yeah, that's a good point.
Mm-hmm.
Yeah, I have a question about the silica microporous zeolites.
Yes.
Because you show that it's not accessible to water. You capture the oxygen, but then how the oxygen is released? And how about the biological fluids? Like, is it necessary to release it into the water? Like, you know,
yeah,
so
in the case of those particles, the oxygen release is entirely passive, so it's driven by a change in partial pressure. So you have oxygen that's absorbed in the particles, either equilibrated in the air at 0.2 bar, or if you're in a pure oxygen stream at 1 bar, then it gets to a low partial pressure oxygen environment, like either tissue that are oxygen poor or in electric catalysis where you've depleted oxygen because it's been reacting, and you have this partial pressure gradient, and so oxygen is desorbed and released until the partial pressure inside your particle matches the partial pressure of the surrounding environment. So it's a very passive release mechanism, which is good for certain applications. It's very similar, or the exact same as how perfluorocarbon emulsions release oxygen. For other applications, you may want to have a more actively triggered gas release. And so there you can think about playing with surface ligands on your particles and using different kinds of triggers to kind of close porosity or open porosity as a way of triggering release. That's not something we've really explored yet.
Okay, thanks.
So is it possible to tune a time scale of release of the oxygen by many orders of magnitude?
In principle, I think it should be. It's not something we've been able to do yet. And so one idea we had was that depending on how polymers or proteins or whatever surface ligand pack on the external surface, if you can make a really dense shell, then you should be able to slow down the release significantly, just like kind of if you wrap your particle in like a membrane effectively. If you have a lower permeability membrane, then the gas release will be slower. To date, all the shells we've made are still quite porous, and gas release is really fast. And so we haven't tried very hard to make it slower, but in principle, it should be very possible. You just need to get the right coating around the material to slow down the diffusion of gas. Right now, diffusion is really fast, and so that's why we see really rapid release.
Are there any other questions or comments? Well, if not, let's thank all our winners and the winner from a previous edition once again. So thank you, Jarad.